I am Christina DeCoste, Director of the Flow Cytometry Resource Facility at Princeton University and President of MetroFlow, the New York, New Jersey Flow Cytometry Users Group. Since we weren't able to meet in 2000 and into, unfortunately, 2020 and 2021, the 15 members of the MetroFlow Steering Committee created this launch seminar series as a vehicle for our corporate members to reach our MetroFlow audience through the virtual platform. You can find our previous online events archived in our YouTube page that you can link to through our website, metroflow.org. You can find us on Twitter at MetroFlowCyto. And if you would like, you can go ahead and subscribe to our YouTube page. If we get 100 subscribers, we can get a handle that's not a long string of letters and numbers. So please do subscribe to our YouTube page. As a disclaimer, MetroFlow remains neutral and in no way endorses any one vendor or their offerings over another. All corporate members have the opportunity to submit a proposal to present in this launch seminar series. And the purpose of these events is to showcase the innovations in the field that we can all take advantage of to better support our scientific and research endeavors. We hope that all of you will be able to take what you learned today from this seminar series back to your labs, and the information will be helpful to all who are watching today and in future seminars. Please use the Q&A for questions. We'll present them to the speaker at the end of the presentation. And before we get started, I'd like to thank today's speaker, as well as you, our MetroFlow members and extended audience, for your time and participation. So with that, I'd like to introduce our speaker, Dr. Kenta Yamamoto, Cell Analysis Product Manager at Biolegend, who will be telling us about their Apotracker reagents. So you can go ahead and share your screen.
And all right, so good afternoon, everyone. I'm reporting from the West Coast, so it's still morning here, but just want to do a quick sound check. You guys can hear okay?
Yes, we can hear you.
All right.
We're seeing your PowerPoint, but not in presentation mode.
Right. I didn't have it up yet. All right. So, well, first and foremost, thank you very much to the organizers for giving us the opportunity to present today. It's truly an honor. And so today I wanted to talk about what I think is going to be a really useful reagent for the field in Apotracker. And as the title suggests, I'll be talking mostly about Apotracker, but I do have to give out to MojoSort as well, which is another product line that we carry for cell sorting. It is related to Apotracker, so I'll be talking about that. So going through the presentation outline really kind of comes into play two different parts here. Really, to start out, I'm going to be talking about what is the now. So I'll give a brief background introduction on phosphatidylserine, which is going to be central to the presentation and what I'll be talking about as an early marker for apoptosis and a reagent that a lot of you, if not all of you, probably know in Nexon-5 base staining, which, as you guys know, is the gold standard for apoptosis detection right now. And then going into the second part of it, I'll be talking about the future, which is the introduction of the Apotracker family. So under that umbrella, we have three products. One is Apotracker Green. This is something that is already out in the field. Some of you guys may be familiar with it, but I'll be talking about that to give a brief introduction on that. And then I'll be talking about the product that will be coming out soon that will be added to the Apotracker family, which is Apotracker Tetra. And then another practical application, if you will, or a product that is useful for, I guess, a practical application that came out of Apotracker, which is the MojoSort dead cell removal that will also be coming out shortly. So stepping into the background section of this, which is to give a little background on phosphatidylserine, or PS as I'll be referring to across the slides that I have here today, and the physiological relevance. So basically, phosphatidylserines are specific phospholipid components that have a serine amino acid bound to the polar head group. And typically in a healthy cell, these are asymmetrically distributed throughout the plasma membrane. So here kind of depicting the phosphatidylserines here in red. Typically the phosphatidylserines are concentrated on the intracellular or the inner leaflet of the plasma membrane. And it's typically kept like this in a healthy cell through activities of enzymes such as flipases that are active in the cells. But what happens is that during apoptosis, programmed cell death, this asymmetry is lost. And what ends up happening is that the phosphatidylserine residues end up being exposed to the extracellular surface of the plasma membrane. And this is in part caused by activities, enzymes such as scramblases, that become activated through caspase activation and cleavage. So physiologically speaking, this asymmetry loss or the exposure of the phosphatidylserines to the surface of the cells actually serves an important or several important cell signaling functions, such as it being a signal for phagocytes such as macrophages to be able to recognize what is apoptotic or an unhealthy cells and serve as sort of a quote unquote eat me type of signal. And another important feature of early apoptosis, at least in the cells that are programmed to die, but perhaps not completely dead yet, is that the structure of the plasma membrane is actually kept well intact. So the structure of the cells and the structure of the plasma membrane and its porousness actually stays relatively intact. Now, taking advantage of the outer leaflet phosphatidylserine exposure, you know that there is a probe called Annexin-5 that's based on the protein Annexin-5, which is now a gold standard for apoptosis detection. So the way that Annexin-5 binds is it binds to the phosphatidylserines exposed in the outer membrane, and when they're conjugated to fluorescent proteins, that these can be used for fluorescent apoptosis and cell detection. And so a typical plot that you might be familiar with in looking at this when combined on a bivariate plot, looking at this in combination with a DNA binding viability dye like 7-AAD, you can distinguish cells that are in early apoptosis as indicated by the Annexin-5 positive cells, and cells that are both positive for 7-AAD and Annexin-5 are really the truly dead and also the necrotic cells. And that's where the component that I mentioned about the integrity of the plasma membrane being relatively intact in early apoptosis is an important one, because they're not as porous and they won't lead to an Annexin-5 binding to the intracellular components during cell death. And this is just an example of what it looks like when you take Jurkat cell lines and treat it with an anti-Fas antibody to induce apoptosis. So you get this nice separation of the early apoptosis and the dead and necrotic cells. So Annexin-5 is, a gold standard for a reason. It's a very useful probe, and many of you guys listening in today will probably be familiar and have used it. But there's a major caveat to the use of annexins, and that is the major one being that they're calcium dependent, in that the Annexin-5 binding to the phosphatidylserines is a calcium dependent reaction. It's known that it requires at least 2.5 millimolars or around that amount of calcium concentration. And what that typically means in sort of a technical or an experimental setting is that you need a specialized binding buffer for it. Typically for regular flow staining, you typically make with regular DPBS. Typically calcium ions and also magnesium and others are excluded from the this regular PBS saline solution. and for good reason. Some of them being that the presence of calcium does potentially elicit artifacts such as cytotoxicity. Calcium is an important key element in cell signaling pathways. So naturally, with the presence of high calcium, it also can elicit that in vitro. And because of that, it can also elicit some cytotoxicity as well and actually cause cell death while the samples are in this buffer itself. And so what that also means is that, 2.5 millimolar is fairly close to physiological concentration, but in certain cases, it may not be optimal for in vivo apoptosis detection through phosphatidylserine recognition. And, you have to give some credence to some of the papers that are out there that do indeed use an XM5 for staining in vivo. That technique does exist, and maybe some of you guys have used it before, but it is also known that, say, for instance, in some disease models, that there is a presence of calcium imbalance physiologically, and that may not be suitable for staining using an XM5, which is a calcium-dependent reaction. So, is there a calcium-independent alternative to an XM5 binding for phosphatidylserine-dependent apoptosis detection? We know that this is a good marker for it, but it would be really nice if there was a probe that can, bind to these things without the necessity for strict binding conditions that makes an XM5 a little bit harder to use in some instances. And so initially, we introduced Apotracker Green. So this is something that we have in our catalog already and associated with the publication. So I'll talk about that initially. So to give a brief background on the molecule itself, it's a small cyclic apopeptide that is synthesized to bind to phosphatidylserines with high affinity. And so this is quite different from an XM5, which is, a full-on protein. These things are, relatively small probes that initially was also designed in, terms of thinking about the permeability in tissues. And another key component about Apotracker Green is that it is an environmentally sensitive probe, in that what we mean by that is they turn on the fluorescence that is quote unquote FITC-like. So it's actually not FITC in itself as a molecule, but it turns on its fluorescence only when it binds to the phosphatidylserine residues. And the developer, Mark Vendrell's lab, have gone through many iterations of coming out with different peptide sequences to really optimize the binding to the phosphatidylserines. And then also the environmentally sensitive component of it is sort of intended to reduce the background signal that would be coming from the probe as opposed to being always on. So, and the most important thing is that this is a calcium independent phosphatidylserine binding reaction. And you can use this in standard FACS buffer, as you guys would probably refer to, or cell staining buffer that's made from DPBS for staining in vitro. So you don't have to use this specialized binding buffer that you guys are familiar with. And here's an example of Jurkat cell staining FACS buffer after being treated with anti-FAS to look at this nice population separation, looking at Apotracker Green here on the x-axis and Helix and IR as the DNA interpolating viability dye to indicate the early apoptosis in the dead and the necrotic cells. So, works very much like an XM5. And then, as I mentioned, because of this lack of requirement for calcium, it can be used in a whole variety of different contexts here in imaging and microscopy. So here I'm showing you some examples of staining that we have for immunocytochemistry, ICC, on HeLa cells, looking at dead and dying cells, as well as looking at live cell imaging, which is a very key component of this. Let's see if I can try to get that video working here. All right. seems like the video was being a little bit finicky, but that's okay. But this is an example of live cell imaging data. And then also, in addition, APO15, which is what the developers had referred to there in the original Nature Communication paper, which is that it can also be used in vivo as well. So this is an example of a staining coming from a mammary tumor xenograft model where Apotracker Green was injected into the mice, and in looking at tissues or mice that were treated with cisplatin, you can see cell death as indicated here, co-staining with cleaved caspase 3, which is another apoptotic marker. So again, demonstrating that Apotracker Green can be used in a wide variety of imaging studies as well. So going from that to the reagent that will be coming out soon from our catalog, which is Apotracker Tetra. And so Apotracker Tetra, unlike Apotracker Green, is actually a protein or a large protein based probe similar to an XM5. So it's originating from the milk fat globule EGF factor 8 protein, so the MFGE8 protein. And physiologically speaking, this protein actually is involved in the process of alpha beta integrin binding. And through its interactions with alpha beta integrin that are expressed on macrophages, it elicits phagocytosis and clearing of dead cells. So what Apotracker Tetra is taking several components that are recognizing the phosphatidylserine residues on this. And another component of this is that the multimerization of this protein actually induces conformational changes that makes it a higher affinity to phosphatidylserine residues. And so what we've done is taken that component that the domains that are responsible for binding the phosphatidylserines, linked it to biotin, where you can tetramerize them using streptavidins that are conjugated to fluorophores, and then forming tetramer complexes, which will then be able to recognize phosphatidylserine residues. And again, just like Apotracker Green, the Apotracker Tetra binding is also a calcium independent reaction, and this can be done in standard FACS buffer, as you can see here. So again, we work with another group that have developed this, and it's referred to as a C1 multimer here, where coming from the developer data here, what you can see here is that in thymocytes that were treated with staurosporine to induce cell death, here what you're seeing is a co-stain with an XM5 and the Apotracker Tetra here in conditions where you have the calcium in the binding buffer, and you can see that it co-stains nicely on the same population. Whereas if you take out the calcium, so this is staining done in regular FACS buffer, that you don't see the NXM5 staining anymore because, as I mentioned, this is a calcium dependent reaction, but you still see the staining for the same population on for Apotracker Tetra, demonstrating that this can be done in standard FACS buffer, and it also co-stains with the NXM5 positive population. Here's some other data comparing Apotracker Tetra with Apo Green. So here the Apotracker Tetra is done using an Alexa 488 conjugate for the streptavidin, as you know, is very similar to Apotracker Green in terms of its fluorescence property, very fit C-like, quote unquote. Showing staining in both the binding buffer and regular FACS buffer, and demonstrating that it does stain very similar populations for both. Another key thing about the Apotracker Tetra is that it can also be used in vivo. I mean, here, what the developers have done is they've taken Apotracker Tetra that was injected in vivo into mice, and looked at sheep red blood cell challenged mice. So this would induce a germinal center reaction and induce class switching and somatic hypermutation in B cells. and will lead to a lot of B cell death. And so they've taken the germinal centers coming from the spleen and then ran them on an imaging flow cytometry platform, looking at CD95 as a marker for germinal center B cells and also, dying cells as well, and looking at Apotracker Tetra staining, and you can see that it co-stains nicely. So what this demonstrates is that the Apotracker Tetra can be used to identify apoptotic cells from mice ex vivo after injection. And looking at GL7 and CD95 co-stains, that these populations that were gated in with the Apotracker Tetra staining is highly correlated with its expression after sheep red blood cell immunization. Another thing that's been also noted in the literature is that extracellular vesicles, such as exosomes, they're also highly enriched with phosphatidylserines as well. So what's been demonstrated is that with Apotracker Tetra, that you can also identify cells that are extracellular vesicle positive. And what's also very clear, I think, looking at this from the imaging cytometry data here, is that instead of these pan-cellular staining that you see on cells that are presumed to be apoptotic, you can see the presence of, in this case, looking at lymphocytes harvested from a mouse model where they were looking at LCMV infected mice. So it's a mouse model for meningitis. And you see these more punctate type of staining, indicating that there are more cells that are extracellular vesicle positive, but not necessarily apoptotic. And so kind of going along with this and talking about what the developer have done is they've actually came up with an algorithm on a convolutional autoencoder, or CAE, that basically was a machine learning algorithm to train and look at and identify, based on imaging cytometry data, that are either EV positive or apoptotic based on the staining pattern. And looking at this in both a mouse model and also looking at patient samples coming from COVID patients, that they've identified that patients that have samples that were harvested from COVID patients, when they ran the imaging cytometry and looked at Apotracker Tetra data, that when they have higher prevalence of lymphocytes that have more EVs, that it's associated with a more severe phenotype of COVID and the disease progression. So this is sort of putting into context the use of something like Apotracker Tetra to identify extracellular vesicles on cells and having some sort of physiological relevance or disease severity associated with viral infections. And then finally, looking at this data for Apotracker Tetra in a model of myocardial infarction in mice. So upon induction of cardiac ischemia, you can see that this probe can be used to detect cardiomyocyte death in the heart, as you can see from the pinkish staining here with the Apotracker Tetra compared to the sham control. And I think this is a good example of data that's been generated for this probe, because I talked about the use of these phosphatidylserine probes in the past, especially with annexin. You can imagine that in these type of models where, especially something like in the heart, where calcium signaling is a key component in cardiomyocyte function, that this probe can be used nicely to detect apoptotic cells in this kind of model, really demonstrating the breadth of this probe in being able to detect apoptotic cells in vivo. So talking about Apotracker Green and Apotracker Tetra, what I've emphasized is that they're both calcium independent probes. And so, they both sound very similar, but, talking about, well, what kind of instance where one might want to use Apotracker Green versus Apotracker Tetra and their usefulness. And I think it really kind of comes down to what's been sort of demonstrated and the thinking about the molecular properties of both of these. So as I mentioned, they're both can be stained in calcium independent buffer and staining in vitro. It can be done in standard FACS buffer or equivalent. They've both been used for extracellular vesicle identification and have also been used in vivo. Another data that I didn't show for both of these is that when you stain the cells with either Apotracker Green or Apotracker Tetra, it doesn't inhibit macrophage phagocytosis. So, it can be used in these type of functional assays as well. I think, the parts where you're thinking about, okay, which one might be useful for my own study, I think here are some of the things that can be considered. So for instance, the molecular size of these, Apotracker Green is a much smaller molecule compared to Apotracker Tetra, which is a complex-based, much larger protein-based complex that is used to detect the phosphatidylserines. As far as the fluorophore options, right now with Apotracker Green, we only have green, whereas with Apotracker Tetra, you can imagine this can be pretty much used with any kind of combination as long as you have the streptavidin fluorophore. From our catalog, we'll have this as a bundle sold with titrated amounts of the streptavidin so that you guys can pretty much put 10 microliters of this, 10 microliters of that, and directly use it. It makes it easy. But if you want to get creative and use other fluorophores that maybe we don't necessarily have a bundle for, that you can also do this as well. In terms of reagent preparation, Apotracker Green is a probe that you can directly use, whereas Apotracker Tetra is a biotin streptavidin binding reaction based, so it does require 2 steps. I mentioned that Apotracker Green is environmentally sensitive and it turns on after it binds to phosphatidylserines, whereas with the streptavidin fluoride, it's quote unquote always on. And another thing about Apotracker Green, and it has been used, is live cell imaging. So really kind of taken together as far as which one might want to use for either one. I think it really does kind of boil down to what might be best for these kind of different instances and what might be best for my experimental setup and what I need. Do I need a particular fluor that I need to use as opposed to FITC or Lexo 448 type of channel for my flow cytometry studies because I already have that channel occupied, that kind of stuff. And, again, we'll be happy to consult with that and help you out with your experiments on that. Moving on from the Apotracker probes to a product that was born out of the usefulness of the Apotracker Tetra. And so what I want to talk about now is the MojoSort dead cell removal kit that we'll be coming out with as well. Very much related to the Apotracker. And so just to give a quick plug on MojoSort, it's our magnetic cell enrichment or cell separation product line. The large proportion of our portfolio for MojoSort is to isolate and select particular immune cells. As some of you guys might be familiar with, these are made with these handheld magnets where you can put a standard FACS tube with your sample, you put it in there 5 minutes, you decant, and then you get your positively selected or decanted out samples that are isolated. And so what we're going to be coming out with is a dead cell removal kit based on MojoSort, where you have a 2-component monomer and streptavidin magnetic bead multimerization that you put onto your sample, incubate, and then you wash out, and then you get your live cells. And the special thing is that this can be done in MojoSort buffer, which is what we refer to, or equivalent, cell separation buffer. And there are no other special buffers needed. So it doesn't require the use of any binding buffers as you would with an XM5 staining. And here's some representative examples of looking at that. So what we're going to have is 2 separate reagents that are optimized for both mouse and dead cell removal. And looking at heat-killed samples in both, where you're looking at starting populations of about 60 to 70%. for both mouse and human kits, you can see that based on the Helix Blue and the Apotracker Green data, that we can get over 90% positive enrichment of live cells from both. And again, to emphasize, this is done in standard cell separation buffer, which, by the way, typically also including our own EDTA, which is a calcium chelator. So, really demonstrating that it really doesn't require calcium in order for it to work. The major advantage of using this type of kit over other ones that are currently existing is the live cell recovery or the yield. So here's some example of several replicates of data that we've generated for both of our up and coming MojoSort dead cell removal kits compared to other ones that are out in the market that are annexin-5 based, which requires the use of, again, the annexin binding buffer, which elicits artifactual effects, and where you see most of this effect is really in the recovery. So you can see here that with both our kits that you get a higher live cell yield, and really demonstrating that you get a better recovery of the live cells, and especially under the context where you might be working with a relatively small portion of the samples and you want to get as much of the live cells out very quickly, then this is a very useful reagent for you to be able to do that. And one particular application that's very much up and coming and is actually very much established now, I should say, is the use of or the live cell enrichment prior to single cell RNA-seq applications. So here I'm showing you examples of samples that are either fresh. This is coming from PBMCs done with a single cell RNA sequencing experiment. Fresh PBMCs, samples where 60% of the sample is viable is mixed in with heat-killed PBMCs, and samples that are taken from this 60% viable sample and then have used the MojoSort dead cell removal to remove out the dead cells. And what you're seeing here is just the percent mitochondrial reads, so genes that are associated with mitochondrial genes, and the sequencing reads that you get for that relative to the total gene read counts. So this is sort of a surrogate marker that is used for looking at the sample quality in terms of the viability. And you can see that when you have a lot of dead cells, the percent mitochondrial reads do really go up. And when you take the sample and then use the MojoSort kit, you really do get rid of this sample, really do clean up the sample nicely to make it really look like a fresh sample. And looking at the UMAP dimensionality reduction plots, this was done in combination with CytSeq, which I'll also mention next, in looking at the different clusters of different immune populations coming from PBMCs. When you don't get rid of the dead cells, you actually do get a cluster on its own. So largely coming from the blue dots here is coming from the sample that have a 60% viability here. And you can clean this up very nicely. You can see very few orange dots on this sample. And then here it's showing the same dimensionality reduction plots based on the heat map of the mitochondrial reads that is shown here. So you can see that basically when you take the samples and run through with the MojoSort dead cell removal kit, that you can effectively get rid of these. And when you do a single cell RNA-seq experiment, it would be very much a shame when you have a certain number of reads and sample to have a lot of this taken up by these dead cells. So you really want to get rid of that. And here's an application where the MojoSort reagent will be really useful for that. Now, looking at site-seq data and the analysis of surface markers on the 3 samples, again, the green is the fresh, the blue histograms that you see across the board here, assessing for the different surface markers here, are coming from the samples that have 60% viability, and then the red is that blue sample with the dead cells removed using the reagent. And then what you see here is that, especially under the areas where looking at the peaks that are circled in red, that when you clean up the dead cells, you do get rid of these artifactual populations coming from the dead cells in the form of getting rid of this high background or dying cells, or in the case when you're looking at something like CD3, the overall, I guess an equivalent of what you're looking at is an MFI for the protein expression, really does go up. So it really does clean up the data very nicely in the context of a site-seq experiment where you're assessing the expression of surface markers using oligo-tagged antibodies. So to go into the summary here, with Apotracker Green and Tetra as alternative solutions to known apoptosis detection methods like Annexin-5, I think that we're really sort of changing the game by introducing these reagents as something that is not requiring calcium, but it works very much just like an Annexin-5. But again, without the use of these specialized buffers, it's easier and better. It's easier to use, and I think is very much better for a lot of the reasons that I mentioned today. And as far as the different types and a variety of different experiments that's been done with these, I think, to kind of go back to that question of which reagent you might want to use, I think it, helps to refer back to that table that I had earlier. I'm sure this presentation will be available to you to look at, and then also to look at the manuscripts to see what's been done. Now, as far as the ETA for the Apotracker Tetra, we're looking at around the mid to end of October. But of course, we'd be happy to help our field application scientist team, who are fantastic. We'll be happy to help you out with your experiments. Our technical team will be happy to help you out, and I'd be personally happy to help out as well. So definitely reach out to us for any sort of technical consultation. If you want a little bit more background on that, on the reagents and stuff, we'd be happy to help you out and help you design your experiments. And then also going into the dead cell removal, we can proudly say that this is quote unquote powered by Apotracker, in that it is a dead cell removal kit that is that doesn't require the use of the Annexin-5 binding buffer, and it's a rapid, effective dead cell removal reagent with minimal artifacts and high live cell recovery. So we'll be updating this, but the place where you can go to on our website to look at our apoptosis and cell health tools, I think the easiest would be to just search that and then go to the apoptosis and cell health tools. We'll have the information regarding the Apotracker reagents, as well as the other reagents that we have to assess apoptosis and cell health. All right, with that, I'd like to thank everybody for tuning in. So here's my contact information. Please do reach out to us, and we'll be happy to talk to you and consult with your experiments. And then also special acknowledgements to the developers of the Apotracker reagents as well. All right, thank you very much.
Great. Thank you so much, Kenta, for that really interesting presentation. We have several questions that have come through, so let's take some time for the Q&A. Evan Jellison asks, with regard to the extracellular vesicle studies, is the Apotracker binding to the exposed phosphatidylserine, and are the EVs distinguishable from apoptotic bodies?
So in those studies, what they've done is part of the earlier figures, and I didn't show this, and I don't exactly recall exactly what markers they've used, but they've demonstrated that the the cells that have the apoptotic bodies are co-staining with particular markers, the CD41, if I recall, to really identify that they're actual extracellular vesicles as opposed to dying cells. And so that was part of the conformation to indicate that these are distinguishable from the dead cells, dead or dying cells, and then use that as a basis to come up with their algorithm to be able to distinguish the two.
Okay, great, thank you. Kathy Daniels asks, since it's not FITSI but similar, do you have information on the full excitation and emission spectra of the Apotracker Green for investigators to reference for cross-laser excitation and considerations with panel design?
Yeah, I believe it's up on our spectra, if I recall, but if not, it should be on the manuscript as well.
Okay, but I would say, yeah, if you could make sure that that gets up onto the Spectra viewer, right, that would be, that's certainly a tool that we use in core facility management, and we refer our researchers to.
Absolutely, yeah, certainly, it's a good point, yeah.
Another question we have is, has there been work to validate that these Apotracker reagents work across species? And are there any species it does not work well with?
As far as I know, that's been used for mouse and human. Beyond that, I'm not entirely sure. So yeah, that's pretty much all I can say.
Another question from Evan. The Apotracker Tetra seems amenable to mass cytometry labeling of apoptotic cells. Is there any reason it could not be used for this technique? For that matter, are there streptavidin RNA tags that could be added for sequencing?
Yeah, for sure. We haven't actually used that technique yet, but the streptavidin PE oligo, for those of you that are familiar or have done CiteSeq and used the biotin streptavidin reaction, that is available. So I'd imagine that's something that is definitely doable. We haven't done it yet. And as for use in mass cytometry, again, I'm pretty sure it can also be used as well with the metal conjugations. With Apotracker Green, I guess it's a little bit harder because it's a probe, so it might not be more amenable to metal conjugation. But no, certainly with streptavidin metals, it can certainly be a feasibility.
Okay, that sounds great. Kathy asks, like cyanine dyes that can non-specifically bind to monocytes, have you seen any non-specific binding of either of these Apotracker reagents to certain cell types?
Not as we know. What we know from product development too is that with different batches of the streptavidin, talking strictly about the Apotracker Tetra in this case, the streptavidin themselves can also elicit some non-specific binding as well. And because they're, a tetramer based on a streptavidin biotin interaction, that this could also lead to some, I guess, certain background effects. But not specifically so much with background binding to specific cell types like what you would see with say macrophages or monocytes, as you would in flow from FC binding, because these things aren't antibodies per se, but not specific. So to answer that question, not specifically with any specific type of cell type.
Great, thank you.
Yeah.
Moving on to a couple more questions on the dead cell depletion. So how large are the magnetic beads, and are they validated to work on cytometers, both analyzers and sorters, after the dead cell depletion?
Yes, so they work after they work for flow analysis after isolation. That's certainly true because we do that routinely for our flow studies. or validation studies to see how effectively we've been able to remove the dead cells, taking both the positive and the negative fraction. The positive fraction being the ones where they're bound to the beads. And then the beads themselves are roughly about 120 nanometers in diameter. So just to give you, I guess, a perspective on, where that sort of falls within the range. Other column-based magnetic cell separation reagents, as you can probably imagine, those are much, much smaller. Whereas things like the Dynabeads, for instance, maybe a lot of you guys might be familiar with them being used for, macromolecule pull down, like IPs and stuff like that. Those are in the realm of microns, right? So, 3 to 5 microns, if I recall. So our reagent falls somewhere sort of in the middle there.
Okay, and have you utilized them with any cell sorters after the magnetic separation?
We haven't specifically done so much of the cell sorter assays, but we know that customers do. And so they can be effectively used. And you can imagine that, I guess with the dead cell removal kit too, that you're essentially collecting the negative fraction, which are the unbound fraction. They don't have the particles on them. So they can be used for that, certainly, because they're untouched.
Great. And the last question I have here, unless anyone else wants to send a few more to the Q&A. With the dead cell removal site-seq data, was it compared to sorting for viable cells using a traditional viability dye prior to carrying out site-seq?
We have not. So that is really the only data that we have so far with the dead cell removal kit prior to the analysis. So I think that's a good suggestion. We can definitely look into that. But part of it also is that, you don't have to put it through a cell sorter, right? And, for the core facility people out in the audience, I'm sorry to say, but, when you put the cells through a sorter, it can, affect the cells just from that process in itself. Whereas the dead cell removal is very much a 5-minute incubation, you decant, and then you're pretty much proceeding on to the experiments. So thinking about how this reagent would be more advantageous over that kind of method, you can sort of think of it that way, in that you're not introducing the whole running that cell through the ARIA prior to the single cell RNA-seq experiment, which can affect reads and expression levels of certain genes, as I can imagine.
Not to mention coordinating the scheduling between my flow facility and the genomics core. So
yeah, sure. Definitely you don't have to do that. You can do that just on the benchtop, or at least in the tissue culture hood.
Yeah, so that's actually some data that I would really be interested in seeing, if you had a chance to do that kind of comparison.
Like a comparison with a viability, that's something that people would traditionally do to
PI or DAPI sort, and then do similar studies to what you had.
Sure, yeah, absolutely.
All right, any other questions from the audience? Right, I don't see any. So thank you everyone for attending. Thank you, Dr. Yamamoto, for your presentation and generously answering all of our questions. This recording will be posted up to our YouTube page within a week. And we appreciate everyone's time and attendance today.
All right, thank you very much for your attention and having me.
Okay, great, thank you.
All right.
Bye-bye.